Ask yourself, have you ever boiled water? You have at one point in your life, even if it was in Cooking Mama on your DS and you were 5. But what do you see when you boil water? You always see bubbles, boiling bubbles. You can always see them. Even if you're just making some good soup, there's little bubbles, pockets of steam that come to the top and hit you in the face. Let's look deeper, shall we? What does this look like on a smaller level? So you can see little pockets or vesicles of trapped steam and the water soup surface. These little vesicles bond with that surface and end up allowing all that steam to go back into the air. This is an analogy for the cellular process of exocytosis, which is the polar opposite of endocytosis, because exocytosis means to exit the cell. Using the correct names, you can see there's a vesicule of molecules, which is either proteins, waste, something that needs to leave the cell, and it actually bonds with the cell membrane, thus pushing it out into the body to use or just into the extracellular space. Exocytosis is also typically used to strengthen the cell, as you can see in this model of the phospholipid bilayer. You can see there's new phospholipids from that vesicles membrane that had bonded to the actual cell membrane. This is not always the case, though. Another method of exocytosis is called kiss and run. It's where the vesicle ends up just bonding to the membrane for a moment and then recollecting itself and leaving. It drops off the materials it wants and then leaves, which never strengthens the membrane and really doesn't do much except for get whatever needs to leave the cell out. Exocytosis is most typically seen being used by the Golgi apparatus, as that is what ships, packages proteins and gets them out of the cell, or it sends them on their way to do so. That's a more common example of a constitutive type of exocytosis, as there are two types, constitutive and regulated. Constitutive is a bit more continuous and a more common type, where there doesn't need to be any neurotransmissions to get something out of the cell. Regulated is a special case where there is a necessary neurotransmitter from the body or another cell that forces the cell to conduct exocytosis. This is seen mostly in neurons and also in the pancreas. When it is conducted in neurons, that's usually the final step in a cycle, which leads to an information leak to other cells and to the rest of the body, typically involving hormones, sex hormones, something of that kind. While in the pancreas, it usually tells the pancreas to give glycogen to the liver and thus putting it into the bloodstream, something to assist the entire body, which is why neurotransmitters are needed for this special case. There is a third bonus case called lysosomal exocytosis, which is a process which leads to the secretion of lysosomal content upon the fusion with the plasma membrane, which rids of waste in the cell and also helps maintain cell fitness. It also is used for cell-to-cell communication, which is very important. Overall, that's everything you need to know about cell exocytosis. Next time you boil water or some soup, try to think about this process and envision it. Thanks for listening.